After the bacteriophage titration, we see that Reed and Munich method of the titration of the viruses. Bacteriophages are those viruses which infect the bacteria. However, for Reed and Munich method, we can just see some other viruses are also those viruses and see them titrations and how much amount of that specific kind of the viruses are present in our dilute in our experiments. So first of all, the titration by the Reed and Munich method. This is also called as endpoint dilution method. Endpoint dilution method is mean that we have already studied that LD50, the lethal dose 50, which is used for the to be used at how much dose is the lethal to kill the 50% of our experimental animals. So here it is called as endpoint dilution method. It is also used to determine the virulence of virus in the animals as an endpoint. Similarly, the serial dilution of the viruses will be used and inoculated into multiple animals which are used as the laboratory experiments, laboratory test animals, and infection of the animals will be determined by their death or the clinical symptoms which are shown by those specific our experimental animals, and these clinical symptoms are included as fever, weight loss, or the paralysis. Different kind of course the clinical signs will be shown by the different kind of the viruses, because all the viruses are specific for showing their own specific kind of the symptoms. So the clinical signs are directly proportional to the virus which we are going to use for some specific kind of the experiments. The results will be expressed as the 50% lethal dose. We have already seen that what is the LD50? LD50 is the 50% per ml or 50% paralytic dose per ml. When the lethal or the paralysis are used as the endpoint, then the LD50 will be used as our experimental results. First of all is the phage dilution, which is very important, or the virus dilutions which will be done. The simple method for determining the 50% endpoints in our experimental biology. Then the concentration of the test substance which we are going to use, the specific kind of the virus which we are going to use, just like as an inoculum that produces an effect of interest in half of the test animals, because it is lethal dose 50. So the half of the test animals will be shown by the death or by the paralytic symptoms. Titration by the Reed and Munich method, different dilutions which we are going to use. Here in this experiment, we are going to use the dilutions of the NaCl, the normal saline, which is also called as saline. So dilution of the 10 raised power 1, 2, 3, 4, 5 will be used, and then the saline as an control, which is without any dilutions. Then the bacteriophage suspension is here on this side. Dilution technique in 4 plate procedures, and the amount of the inoculum is of course the same. Here is the control and the inoculum is the same. Maybe taken is it compared, it is dependent on the concentration of the inoculum or the concentration of the virus, that how much lethal is the virus is, and then we can just use it for the Reed and Munich method calculations. Here are some special kind of the terms which are often used for the Reed and Munich method. Before you need to calculate the Reed and Munich method calculations for the virus titration, you need to see these terms and you need to understand these terms very accurately. First of all is the LD50. We have already studied it. It is also the abbreviation of the lethal dose 50. The 50 of our experimental animals need to be paralyzed, need to be show the clinical signs, or need to be died for calculation of the lethal dose 50. Then is the effective concentration 50. Of course, the concentration of the virus is very important. Concentration of the virus in the inoculum is very important. So similarly, as far as the concentration, the effective concentration 50 will be important for using the Reed and Munich method. Then the IC50. IC is the abbreviation of the inhibitory dose 50. So not the inhibitory dose, but the inhibitory concentration. The concentration, first we see the effective concentration. It's the other one is the inhibitory concentration. Then the TCID 50, tissue culture infectious dose. We are using the Reed and Munich method for two different ways. One is the tissue culture and one is the in vivo with the experimental animals. So if we are using with the experimental animals, then it will be different, and if we are using in the tissue culture technique, then TCID50 will be very important. Here we just take an example of the read and munit method. We will see in other slide, the next slide which is coming, that how to calculate the virus titrations with this example. First of all, the lethal of the poliovirus we have seen in this example, and in the mice are the experimental animals. A total of the 8 mice we have taken for this experiments. Those experimental animals, those experimental mice were inoculated for virus dilution, and the endpoint was of course the death for this experiment. The statistical method of read and munits was used, and we have determined about the 50% was the endpoint. The results are pooled in the tabulated form, and we can see it in the next slide, and the mortality at each dilution was calculated. The endpoint was considered as 50%, and the endpoint was in between the 5th and 6th dilution, which is calculated as 10 into 6.5 to 10. So of course the resulting virus samples will be 1.6.5, and it is termed in the form of the LD50 units as a 100.5. We can see it in the table. Next slide, that how we put it into these calculations. Again, to describe the details, the table is quite clear, and you can see the dilutions which were used. Of course, the number of the mice we used, the total number of 8 mice, so we can see that how much mice were died and how much mice were alive. First of all is alive in this concentration, in this dilution, these are the alive viruses and these were the died viruses, and then the total alive, total cumulative, and the total dead, and then the mortality ratio, and then the mortality at the end, and then we can put it in the read and munich formula. First of all, the dilution was this. In this, the total live was 0. All of the viruses were died, because this proved a very strong dilution for the polioviruses in those mice, and then all were dead, of course. Then the total live will be the 0, of course, and total dead will be the 40, because we are using it into here the 50% end node. So after the experiment, we come into conclusion that 1 into raised power 6, just to conclude, 1 into raised power 6 was the 50% end point, and for calculation this we come into here, and then we calculated it, and it come just like we have given in the last slide, that we come into, after putting it the formula by through the read and munit method, we take it as 1.1006.